Okay, good afternoon, everybody. Please take your seat. So it's my great honor to welcome you here at Semmelweis University. This is the second Biomedical Data Science Summer School and Conference. My name is Ron Molontai. I am the head of the organizing committee and also the head of the Institute of Biostatistics and Network Science here at Semmelweis University, the institute who is organizing this 2-week-long event. So this is an open lecture, a special open keynote lecture of this 2-week-long event, the second Biomedical Data Science Summer School and Conference. I'm emphasizing it because many of you are just here for this open lecture, but who knows, maybe next year you will join us for either for the summer school or for this 3-day-long conference. Without further ado, I would like to introduce our afternoon keynote speaker, Professor Andreas Dengel, because you are here to listen to his keynote lecture. And this introduction will be not the usual one. I won't read out his bio, but rather I will show you or present you a short video that we created to introduce and welcome Professor Andreas Dengel. So now I will turn off the lights to create a little movie theater atmosphere, and then we will watch the video. So please join me and welcome Professor Andreas Dengel.
It's a heavy burden. It's made by AI, not by me. So thank you very much for this kind introduction. I'm overwhelmed by the audience. Thank you for coming today. And please don't make pictures. I will hand over my slides to the organizers. So if you're interested, you can have a look to them later on. So could I see where I am maybe here on the screen as well, as I did before? Is this possible? So today I will give you some perspectives on what we do in research in the biomedical field. To be honest, I started to dig into this field about 7.5 years ago. Before, I only had half a year of biology at the high school, where I focused more on physics and on math. And today I'm working at DFKI. May I ask you who knows DFKI? Hopefully now some people more. So DFKI is a research company, first of all. So I'm not only faculty, I'm also part of this company. And this was an experiment starting in 1988 by the German government. I have to stay here, sorry, because of the camera. By the German government to not found another Max Planck or Fraunhofer Institute, but to really encourage the German industry to collaborate in AI. So 1988. And since this time we exist and we have no basic funding, meaning that all we acquire is related to projects. The very the German common suspect companies, which you can see on the top right on this chart here, on the slide. We also have international companies as shareholders, as owners, like NVIDIA, Microsoft, Google, and Intel, and some more. And we are independent, so all of them have the same share, 1/33rd part of the whole share. and we have to convince them to give us contracts. But beside that, we are working with almost every big company in the world somehow, including also companies like Apple or Sony, NTT, NEC in Japan, and much more. And as you can see, we have many employees. We are the largest independent AI research institute in the world. And many people from all over the world join us to be part of our team. And beside the headquarters in Kaiserslautern, where we also will install a collaboration very closely with Semmelweis University. We have many sites in Germany and many ongoing projects, which are, of course, not only in the medical domain. So coming to the main area, I cannot go deeply into that, but I give you an overview what we are doing. We are, of course, also working in various other domains in medical part, whether it's in dermatology or in ophthalmology, in cardiology or in radiology. There are a couple of papers mentioned here, which I will not address specifically, but we are also introducing very innovative explainability methods to not only make a decision or propose a decision by AI, but rather also to explain, to justify why this decision is maybe valuable. Moreover, we have a very close collaboration with a big German company called Sartorius. Not sure whether you are familiar. And we are working in cell culture analysis, mainly also in the domain of segmentation. First of all, we try to overcome the lack of data, and we produced a very widespreadly used data set called LiveCell, which is open source, and many people use it. And beside that, we also worked on generative methods to produce synthetic data with a quite good accuracy. At the same time, we tried to be very general in segmentation, and we developed a very robust segmentation technique, which we published last year. It is already cited almost 400 times. Gives you an idea how good it is. And I think I saw some methods this morning in segmentation. Maybe the method also would work for other domains. Beside that, we are also working on 3D cell segmentation. So if you have a volumetric image of cells, which helps you to overcome the bottleneck if they overlap and interact somehow where the shapes are not clear. So the main issue here is that we used a special approach for 2D image analysis, where we layered the entire volume and then could reduce the compute time to only 9% of the original compute time necessary for the data. And this is a quite big innovation. And finally, we also do cell co-culture segmentation and tracking in order to measure the vitality of cells to get an idea about their behavior and how good they are maybe for certain therapeutics. Today, I want to focus, maybe I have no image, I have to resize the one here at my smaller screen. Today, I want to focus on technologies that are addressing the entire drug development process, where we go from the target identification at the beginning to screening and optimizing of the compounds, to find the lead compounds, to evaluate those, and finally also to address the topic of drug repurposing. And I want to show today how AI could help to push these things forward. So the main field I want to address is today the sequence analysis domain. Sorry. And where I like to show you how we can use various types of input from DNA, RNA, and protein to do many tasks. And if you look at the field in genomics and proteomics, we have typically these three types of tasks, namely classification, clustering, and regression, where the classification to primary forecast or just split the various samples in different parts. In the clustering, we focus on the grouping, which elements are quite similar. And here we estimate some numerical values, how they are progressing in the future. And if you look at the various fields, in DNA, you can see there are a multitude of different tasks. Gene function prediction is a typical multi-class classification task, yeah, and there are many more. So, and the same holds for the field of RNA and also for the field of proteomics, where we have proteins here with a quite huge bunch of various questions which arise, which might be addressed by the different wet labs and the research groups. If you want to know more about this, I also like to refer to one of our books, which was published last year at Springer, which has a kind of introduction to this field, but also an advanced part. I come to that later. Anyway, if you look at this field after data collection in any genomics and proteomics sequence analysis task, data processing is an important task in which usually sequence samples with different lengths occur, and finally they have to be standardized. And typical methods, for example, are this padding and truncation, yeah, to standardize the lengths. And there are various techniques to pre-process the data. And after pre-processing, we have to transform the sequences into feature vectors. And there are numerous methods of which you might know already. But we also have to identify the relevant features here through the feature engineering part in order to prepare the data for classification or for prediction tasks. And after prediction, yeah, we do usually this performance evaluation to see what's best might fit in order to go to automation. And we have worked for a certain time on these various steps and looked into literature to establish an entire framework which allows us to really have a pipeline of processing where we collected all relevant pre-processing techniques in one tool and at the same time implemented statistical means for representation of the sequences, but also looked for which language models are valid for them. We analyzed 700 language models worldwide and selected the best 120 of them and put them into this pipeline. And moreover, we have different feature generation techniques and the predictors, the evaluators. So if you combine all these options you have, there are more than 20 million options. And this is just for experiments, because at the beginning, if you do experiments, not in the wet lab, but in the dry lab, you also have to get an idea what might best fit here. And we also compared this framework to existing frameworks, which you might know, iFeature, for example, or this BioSig Analysis 2.0. We have the iLearn and the iLearn Plus, and also the math feature. And we can also address in our framework all the various processing steps, where we offer a rich set of alternatives which can be combined for the analysis. And to get an idea how this works, we also put this into some experiments where we selected some use cases like histone occupancy prediction up to this protein interaction prediction, and looked to the best classifiers in literature, which we tested against a huge data set we have. I'll come to that later. And then combined it to the options we have with the SEC Studio. And you can see that we were better than the state of the art. Some of them is only close, but some of them there's a big distance here to be done. All of this are published in papers, are also included this morning the references. So if you are interested in this specific use cases, you might also have a look into the papers. The second part I like to address is biomarker identification, which is a widespread field. So if you look at the biomarkers, there are many different types in general. Biomarkers are diagnostic or have the task to monitor. So there are many distinguishing aspects here, and each serves as a representative of different purposes. For example, diagnostic biomarkers help to identify diseases. We have monitoring biomarkers are used to track patients' health over some period of time, and others have their own specific applications and contents. Now, when we talk about biomarker discovery, one of the key challenges here is to be general. Yeah, so this means a biomarker that works well for one group of patients not necessarily also works for another cohort. So the idea here is that we identify very universal biomarkers that could be a kind of lead in a certain field of diseases, and is also robust if you would apply them across multiple populations of people. So in this context, we are collaboration with colleagues at the Johannes Gutenberg University in Mainz, with whom we are part of one of the future clusters in Germany, where we focus on cardiovascular diseases, especially also on biomarker discovery. In this slide, you can also see that we are able to classify the vascular narrowing or the severity of the conditions into high, medium, and low categories. And in our study, we investigate how certain clotting factors, F7 and F10, in immune cells, so myeloid cells, influence the development of arteriosclerosis. In these pictures you can see over here, you can see aortas stained with oil red O, showing that when the gene for F7 is knocked out in certain immune cells, we see less arteriosclerosis, which has a protective effect or impact on the vessels, whereas the myeloid deletion of F10 accelerate progression of arteriosclerosis, thus producing exactly the opposite effect. These phenotypes were associated with preserved vascular flexibility and pronounced stiffening of carotid artery, respectively. Yeah. And in order to do that, we also did a special tool called explainable, which is quite similar. It's a pipeline, and this pipeline is accepting multi-omics data, so 8 different types of data, and then applies data fusion techniques, infusion, late fusion, early fusion techniques, which can be combined as well. And then we try to do data reduction based on the huge amount of data to apply classifiers. Also, regression is possible, and then do evaluation. And at the same time, we try to implement various techniques for explainability to get a better understanding why these biomarkers are selected before we again do an evaluation. Here you may see the various biomarkers that we have already identified. These biomarkers are associated with these 3 categories I mentioned: high, medium, and low. And the purpose of this slide is to visually illustrate somehow the results of the framework. I may also refer to a large-scale analysis on biomarker discovery, which I mentioned here on this slide, where you could have a kind of landscape overview what's going on worldwide in this field. So the third part of the talk is related to a knowledge graph. And this was a really hard time for us because we found out that worldwide there's a lot of data existing in the biomedical area, but it's very distributed. It's heterogeneous, no clear terminology, etc. So we put a lot of efforts on that to develop this very unique source of knowledge. And at the core of biological systems, genes, proteins, metabolites, and a wide range of other molecules orchestrate the complex activity here. And these activities are very depending on each other. So if you change one pathway here, there's a whole effect of other pathways in the body. And this means that there's a big need to get a better understanding of the biological effects and dependencies in our body, in the subjects which have individual influential factors, which are not only coming from inside our body, but also via the food or the living conditions, etc. So the idea here is that we want to get an insight into the various types of correlations and model them, because if you have insights, for example, in metal area, into genes, enhancers, transcription factors, RNA binding protein provides basic basis, or provides a basis to do many things, including new therapeutic targets and so on. But this also holds not only for the medical part, because these biological relationships also exist for plants. So you can do also a lot in the field of agriculture, or even in the field of nutrition, where the industry is also looking for new types of fermentation processes, to build more healthy food and so on. So there are certain demands, and we are already collaborating with one of the major European food producers to do such things. So as long as we want to understand these biological processes and pathways, we collected worldwide entities from more than 1,000 sources. And these are widely distributed, very different in their quality, as I already mentioned. And we put them together in a huge knowledge graph where we have nodes like biological entities, RNA, enhancers, promoters, whatever is necessary. Even diseases are part of it. And the edges address their correlations, their relationships. Maybe it's an association only, but maybe it's also a very closer, explicit causal aspect, which is validated through experiments. So everything inside this graph is validated and is present in literature. At the end, this knowledge graph includes more than 650 million triples, meaning that you have a head, an association or correlation, and a tail could be in head again. So you are mixing up all these biological relationships in one big graph. And there's also a paper I'm mentioning here, which gives you an idea how to work with it. And later I give you a link where you can play with the tool by yourself, to just get an idea. So we included sample node of various types. And the statistics are that we have more than 4.5 million open reading forms, about 2 million genes, about 1.6 million diseases, and so on. And also to give an idea about the relationships, there are relationships like cell line, gene expressions, or mRNA protein interactions, and so on. So in total, there are more than 200 types of relationships. And to give you also an outlook to the future, we are also doing this right now for chemical compounds, where we, in addition, collected more than 200 million triples, which will then complement this from the compound side, to get the full picture of what could be done. So Also to explain what we did in this context. So when we looked for the data at the beginning, we did it manually. Nowadays, we do it with a crawler, which is automatically screening the web and getting all the new data. And we have connectors to transform the data into our representation. But we had to deal with inconsistent identifiers. Sometimes even the identifiers were missing. We had mixed sources where different species were present. And we have missing representation of sequences as well. And in the graph, we have explicit names of all the entities, which was a bit tricky because sometimes we had to do subgraph analysis to identify the relationships to come to the conclusion what it is. And we organized it also specifically to the various species. And in addition, we included the sequence and composite structures to get more comprehensive information. So in general, there is a graph of graphs. Depending on the use case and your demand, you can also have a certain perspective on the graph, including also the transcriptomic data sets, which are relevant in some cases. And you could build applications on top of the graph. You will get an idea through this bind tool, which I'll show you later on. Coming to a more concrete application here, namely the gut-brain axis, where the idea is to explore pathways which are not yet discovered. And we are using a part of this graph to give you an idea. Here we have a subgraph, which is composed of more than 580 million nodes across 16 biomedical entity types with more than 3.5 million edges and more than 100 relationship types. And the idea is here to explore the diet, microbiome, brain disorder relationships. So the entity types are chemicals, proteins, as you can see, pathways, microbes, and so on. And for the edges, it's mainly addressing the bodily system or molecular interactions. Some examples are shown over here. In order to give you an idea how this works is, if you look at this microbiome, which has a quite lengthy name, I skipped that. This is producing a kind of plant. What is it? A plant. I think it's a pigment as far as it's quercetin in our case. And this is a flavonoid, which could be found in food like red wine, onions, green tea, apples, and some more. And this flavonoid is then interacting with aromatase, which is an enzyme responsible for converting male hormones into female hormones. And again, this interacts with choline. And choline is a special natural nutrient, which is also present in food, but sometimes also is used as a diet supplement. And this again is then helping to understand through which pathway you could deal with autism in this case. And this is just one pathway of many. Of course, in the world, there are many researchers which are already working on this and detecting some of the pathways, but there are millions, maybe hundreds of millions of pathways which have not been discovered yet. And to do this experimentally, you can imagine that it's very time consuming and cost effective, not effective, ineffective, and maybe causing time of years or decades to detect them. So the idea is that we use AI algorithm that we can use to train a model with the already known relationships. This means you can also think here like a binder problem, that we can train it twice. Here, the idea is to detect by the already known relationships, the unknown ones. And to do that, we are employing the knowledge graph to build a gut-brain axis link predictor, of which you see a high-level overview here on the slide. Based on a given task, you might be able to select a certain microbiome. On the left-hand side of the slide, you can see it. In the input graph, this microbiome is connected to various neurons, to cells, to diseases, to biological activities, but there are missing links, unknown connections that finally we want to detect or discover. And our objective is to predict these missing links and complete the network as shown on the right-hand side, where you see the new links with certain probabilities, which can be derived via this graph neural network, which is shown in the middle. So now conceptually, as I told you, it's like this binder prediction task, where usually we have this kind of head relationship and tail, and you can train it in two ways. Either you have the head and the relationship and predict the tail, or you have the head and the tail in order to conclude on what relationship might be the best one here. To give you an idea, so we also benchmarked our model against several existing methods. Our model is on the left-hand side of the slide, and we used a huge data set for that. And I'm happy to share the results with you, and you can see that we reached about 99% accuracy here across different tasks, and this suggests that our predictor is highly reliable for discovering new connections, which can then be experimentally validated via our colleagues in the wet labs. We also developed an additional module within the same, using the same predictor here to identify biological pathways. So what I mean is that not just single connections, but entire chains of connections or interactions are identified, which are linking microbiomes or microbes, food, and also brain disorders through intermediary entities. And the resulting paths are ranked, as shown on the right-hand side here of the slide, and can be explained by the system. And this is also done here via an application we have built. So to visualize the thousands pathways in a different way. So you are able to select one disease, and the system is giving you an idea about what chemicals, microbes, and other biological entities are connected to that. And you can then go for the experimental validation of this to see what's possible. Finally, I'm addressing a field of application which is growing a lot worldwide, namely this drug repositioning or repurposing domain, which was valid also through the times of COVID, where people tried to react very fast and found some existing drugs which could be applied also to help overcoming COVID for some time. So we can see that it's important based on several dimensions. It's important because usually the introduction of a new drug is time-consuming. As you are experts much more than I am, you know that we need about 10 to 12 years, depending on the various aspects here. And many diseases still lack ineffective treatments. There are many rare diseases for which it's too cost-intensive maybe to develop new drugs. So it might be valuable to see what existing drugs might help in such domains. And we also have this untapped potential of drugs. And the opportunity here is that we might identify new fields of use for the approved drugs, leverage existing safety and clinical data, and reduce time and cost to reach the patient. And AI provides these options to employ this multi-omic data to do it at the same time, and also learns this biological networks from which I gave you hopefully some idea what is possible, and also predict finally the relationships, maybe not only in a positive way, but also the side effects are important to be considered. And at the end, there are a couple of impacts with a positive dimension. So in this respect, we developed this system called COMIC, and COMIC attempts to give not only a pathway from a relationship of a drug to a disease, but also an explanation why this is. And maybe I start the movie and hope it runs. Oh, I'm not on the slide. Now it should run. Yes. So what you could do is you have a couple of buttons here. You can see indications. You can just click on them, and then you have the options to select from the 1.6 million diseases you have in mind. And I selected Alzheimer here because it's common sense. And then you can get a list of drugs which might be relevant with a certain score, which is calculated by the system. I'll let you know in some seconds how this works. And you can also modify the total number of paths and also the length of the path, the top K path candidates. And then the system is calculating these results and comes up with the proposed pathway. To remind you, it's contraindication. And then you have the insights here. You can click on the paths and you get an idea that there is this drug we selected, and then there is a relationship I can't read from here to some maybe protein, and the protein again has a contraindication with another drug, and so on. So the system is giving you an idea how this pathway is valid in this respect. In order to see how good this works, what happens now? I have a problem, sorry. Yes, sure. Want to switch to the next slide? Good question. Me neither. It's an endless loop. Oh, no, now we are here. Okay. Thank you. In order to get an idea how good the system is, we just selected the latest approved drugs by FDA from last year, and we put in the disease as the input, and then you can see this is the approved drug, and you can see on the right column here the probability which our system produces that this drug might be valid to overcome this disease, or at least to help in this domain. So we also published this. I think I had, yes, the reference over here. But anyway, you can also imagine that the system is producing many hypotheses. Yeah, maybe 60, maybe 200, depending on the case. And this is still too much. And in order to reduce this, we want not only do a quantitative analysis, but rather also get a better understanding which of the pathways might be the best one. So we asked this question, how to reduce this number. And therefore, we did this qualitative analysis, combining the big knowledge graph we have together with the existing literature, which is a challenge because in 1 year, there are more than 1 million papers in this domain. But anyway, we got aware that in these papers, there are many hidden treasures, because usually if you do your medical doctor, you do a lot of experiments having a target in mind. And there is a side effect. Maybe there are some results which are not directly related to your target. So they are hidden somewhere in your papers. And that's an insight which we found. And so these undiscovered relationships even play a more important role. And there are some other motivational factors over here which are important. And therefore, we are addressing this interdisciplinary scope by this combination I already mentioned. So we trained a large language model on the literature of the recent years completely. And then we employed the trained large language model to transform every single graph node into a high-dimensional vector. Yeah. So we received this vector representation, and then we calculated the semantic similarity of any two nodes in this high-dimensional space. Yeah. And then we were able to do experiments with two nodes we select in order to get an idea about their relationships by using a cosine measure in this space to get a kind of idea how the distance is and how close they would be relevant for identifying the most relevant path at the end. So in the first step, we received this most relevant path out of the proposed path of this comic system. And then in step 2, we are using a agentic framework, which we have developed, where we have different agents. The first agent is a kind of medical specialist for any type of field or any field here. And he is doing a detailed analysis about every single relationship of this path, looking for the medical background to justify why this makes sense. So it gives a kind of opinion, yeah, to a second agent, which is a scientist, which takes this input to formulate a scientific hypothesis, which might be the basis for a clinical study or a preclinical study. And at the end, we have a kind of a reviewer who is evaluating critically whether this all makes sense, based also on the norms and the laws which are given, and then gives feedback. And they are negotiating all the time. And finally, they come out with a report. And this report is about 12 to 20 pages. Most important is, of sure, the prompting, so which we developed, and also which is on demand generated spontaneously by the agents between them, depending on the mechanism you provide to do so. But you receive at the end something really valuable. We handed over these reports to medical experts, and all of them very much confirmed that it makes a lot of sense to proceed like that. And this gives you also an idea about the power of this AI in general. So if you are more interested, as I promised, there's a website, OpenDFKI, where not only these tools are presented today, but there are more tools you can just use for your own purposes. You can upload your own data, make your experiments to get an idea about the quality, maybe non-quality, depending on the use cases. We are happy to get your feedback on that. And if you are more interested in the principles here, just want to give a hint to the 2 books we published last year. where we collected all the insights we received while doing this research. Thank you very much for your attention and time. And now it's time for questions.
Indeed, we have time for questions. Professor Fenio.
Thank you. Thank you. Very, very interesting and lots of food for thought. I wanted to ask about the knowledge graph. Two questions about that. One is, how do you handle data quality? Different data from the literature have different quality. Is it even possible to address that? Or do you include everything?
As far as we understood that if you have serious and verified experimental data and you publish it, you have to send it to an organization where the data is collected. And we only use such data that is verified through experiments to make sure that there is a solid basis for the data.
Okay. So the other questions is about if you're interested in a specific context. So I assume that, for example, there's a lot of data coming from cancer, but let's say you're interested in adverse pregnancy outcomes. So can you focus in on that?
Not really.
No.
Yeah, we can, of course, open up a perspective through the input. So if you have a certain demand and you want to analyze certain aspects of biological relationships coming from a specific field or use case, you can request or query the graph respectively and see what comes out. So we are would be very happy if you have such use cases and we can work together, maybe to solve problems, because we are lacking in experience in the medical fields. We are AI guys, so we are depending on you.
Thank you. Yeah, we don't have a good solution yet for that. So it would be great to collaborate.
We have the tools.
Thank you.
And you have the problems, and maybe we find solutions.
Thank you.
Sounds like a beginning of a beautiful collaboration, right?
Exactly.
Okay, any other questions? Barry.
Hello, very amazing lecture. My question would be that, especially in the EU, AI is very heavily regulated, especially with the AI framework or AI risk framework, especially applied to medical data. And how do you deal with that? And what steps do you take, especially in a framework that is such on a grandiose scale and it has many steps that has to be top quality?
Absolutely, very good question. And this not only holds for the EU, we have similar frameworks, constraints in other countries. We, for example, are working in a very huge project with colleagues in Japan, where we have access to 10 million patients from hospitals. And here we have to really take care on anonymizing the data. And some of my PhD students are working on such topics, how to transform this data in a representation that would not allow to reconstruct the data anymore. And there are very promising results here to do that. And hopefully the government would be convinced that they could provide the data and we take care that we do not violate privacy aspects.
Thank you.
Okay. Leah?
Yeah, thank you very much for this very interesting talk. I would also have a question for the data you crawl, because all of the literature is heavily biased towards positive results, right? So all of your knowledge graph is identifying associations, but not necessarily if there's not an association, right? So especially for your missing link prediction, how do you deal with that kind of bias in the data?
Yeah, one of my PhD student is trying to find out all the negative mentioned aspects of the experiments. And try to transform them into a representation that could be used in addition to what you mentioned, but that's absolutely correct. Yeah. But we also already identified this weakness, and we try to overcome by a systematic review of what's published.
Any other questions?
Same holds, by the way, by results which could be proved or could be seen as not valid anymore. There are some papers also showing that these experiments have a mistake or the process was not the right one. So we're also trying to do that.
Joby, go ahead.
Okay, so if I understood correctly, the knowledge graphs are species specific, right? So, and what do you think about the idea of connecting them between species, like who eats who, something like that?
You could.
Yeah.
This is not an exclusive thing. So we have the entire knowledge graph. You could have this specific view for any type of species. Yeah. Same holds that you want to focus on protein-protein interactions or something else. So it allows you to also have a specific view to the graph.
Okay, thank you.
Nicole?
Thank you. And thank you for coming here and thank you for this lecture. My question could be a bit off topic. I am curious about your opinion about like where the human factor is involved. Like as somebody, a layperson would assume that your research group is just an expert of everything from gut-brain axis to cancer to like everything. Like, okay, my question is that do you think it would be necessary to introduce the very human feature of thinking outside the box into some sort of predictions? Like AI could sweep every square millimeter of the box, but still within the box. And what is your opinion on that? Thank you.
There are two dimensions. On one hand side, I told you that we are working really in several aspects on explainability. So the system has to justify. But the main driving factor of this research is to reduce complexity, meaning to save time, to be more precise by narrowing down the number of hypotheses, by making them more valid for the wet lab, because the final decision, also the final, say, in German, I know what to say, but in English, maybe to make this kind of nice treasure on the cake. Yeah, this is the task of the human at the end. So the human in the loop is quite important in all risky domains. Yeah, but this tool helps you to understand those patterns which are invisible to us.
Just thank you. From my own experience, I know and I see that this is some sort of an experience of the world that even researchers, like everyday people, tend to, with a sigh of relief, rely on AI. Okay, so now I not only save time, but the effort of thinking. So what is your like experience with that? Like, I think even seasoned experts can tend to take the path of less resistance.
You're absolutely right. I think one very obvious example is the GPS in the car. Yeah. So while the early humans, they used the landscapes for orientation, Yeah, nowadays through GPS, we are losing everything because we just enter the target and then the car is going and we're just driving that way. And humans tend, yeah, if something works, to accept, yeah, avoiding to think. And it might be the task to set or to put AI in the role, yeah, to really encourage not only or to animate humans in a certain way to think about the solution. So also putting questions in addition to what is produced, yeah, to give hints where to verify and how to look at it. Yeah.
Great. We still have time for questions. The gentleman here.
Very impressive lecture, and I have one project. It's about the graph IEG, and I'm also constructing the graph knowledge database. I would like to ask how to define it is a perfect graph knowledge database.
It's never perfect. Yeah. So it's a collection of known relationships. But as I told you, there are many unknown relationships in this graph, as shown in this example of the gut-brain axis. And for every domain, it's the same. So the idea here is also through this drug repurposing to identify new pathways. I simplify the examples to understand it, but the graph is here that between biological entities, there are missing links. And through various applications, we can push AI to use the graph in a way that it forecasts maybe new relationships which might fit to step by step complete it through the verification in the wet labs. Yeah.
Okay, thank you.
Yeah, Adam. So that's the last question that we have.
Thank you very much for the great talk. I was wondering, you framed this drug repurposing as an explainability problem. I was wondering, we tend to see this reverse engineering of syndromes based on how different drugs work and act on different systems or organ systems in the body. And I was wondering how to overcome the possible skepticism from the medical field and the clinical field towards our models and towards these model outputs that we see, these scores that maybe these seem reasonable, but still how to get to the translational part.
I think we are now exactly at the point that the abilities of AI are getting such a big power that we can see that the quality of the results in many cases are better than the ones of the experts. Now, if you look at dermatology, there are many papers proving that the classification of lesions, for example, on the eye side could be better done than by the human experts. And this is true for many domains, but we only could overcome the hurdle that this technology is accepted by proving its value. This means that we have to use this as a tool, assisting system, yeah, which could accompany the human expert by helping her or him find the right way. And if this is verified, step by step, this technology will be accepted. I'm pretty sure we already have some experiences on that we had very convincing use cases, very narrow fields, but anyway, with a quality that was producing some wow effect.